Hi, I'm Dr. Patty Ferris. I'm a board-certified dermatologist, and I'm the co-host of Skincare Confidential. So I'm really excited today to have a good friend of mine, as well as somebody who really is a legend in the skincare business, and that's Dr. Karl Thornfeldt. I'm going to read his bio because it's fairly prolific, and I certainly don't have time to memorize all this stuff, but you're quite accomplished. Interesting, because I always learn so much about people who I've known a long time, which Karl and I have been friends probably, 25 years?
Yes.
I think so. We share a love of skincare, which is how we met years ago at the AAD. Anyway, you received your medical degree from Oregon Health Sciences and completed a surgical internship at the Naval Hospital in San Diego. You were then stationed in the U.S. Navy submarine base in Scotland and practiced as a GP there for 3.5 years. You came back to the States and did a residency in dermatology then at UC San Diego and moved to Southwest Idaho, where you practiced as the only dermatologist for 10 counties spread over 30,000 square miles for over 30 years. That's a story in and of itself. Unbelievable.
Patty, it was only 110,000 people, so it wasn't quite so bad.
The geographics of it, just that alone, that's awesome. You have 22 patents that were granted to you. You've published extensively scientific articles, chapters, and textbooks, but most of these have been on cosmeceutical delivery systems, natural products, and unique dermatologic therapies. In addition, you published a book, which I was not aware of, The New Ideal in Skin Health, Separating Fact from Fiction. And you spent 35 years researching abnormalities in the skin barrier, delivery systems, herbals, and topical therapies for skin diseases. You founded Epionces Inc., and you are the producer and the worldwide distributor of Epionce skincare line, which is what we're going to talk about today. And after years of skin barrier research and really seeing firsthand on your own patients with skin diseases, when you developed your skincare line, you quickly began to see how much of a difference this made in your own patient population. So with that, I'm going to welcome you to Skincare Confidential and tell you how excited I am to hear about your journey. Yeah, it's going to be so much fun. So Epionce is really your baby. You started in 2002. You were way ahead of the curve back then. You were talking about the barrier, honestly, before most dermatologists were really talking about the barrier. I know you work with Peter Elias, who was, again, another legend in this area. So tell me about how that sort of guided you into developing a skincare line in case you didn't have enough to do because you were taking care of all these patients.
Right. Well, the good thing is I never required much sleep, Patty. So that was excellent, made it happen. But one of the reasons I didn't require much sleep is because I had bad atopic dermatitis when I was a child up through the teens and intermittent episodes of exfoliative erythroderma. Even though my dad was the second board certified allergist in the state of Oregon and was chairman of pediatrics at the med school for a while, in Portland. Even then, I was in and out of the hospital and on various kinds of medications that resulted in long-term effects. So I was very passionate to find out the underlying foundational problems. I was actually mountain climbing to relax one day, and I was on top of a 9,000 foot peak and noticed the difference in the environmental effects, the way the sun felt, the wind felt. And I'd been doing all this research with Dr. Stoughton in topical steroids, topical clindamycin, and even some work with retinoids and so on. And it just struck me that everything we were doing was actually damaging the skin barrier, thinning the skin barrier. And it struck me the real true purpose of the skin barrier is protection from the environment. And so then, as I pondered this problem, and I had been doing research in lipids, I decided that we needed to have a group that just focused on epidermis, from stratum corneum to basal membrane zone, and really understand. And I think our biggest discovery, and I founded this group with Peter Elias, a true giant in our industry, and because he was one of the only other people doing any lipid research at that point in time. And so we put resources together and used everything I had financially to help start this company. And then we did 9 years of basic research, and we found several major discoveries. Number one is that the stratum corneum was actually a regulatory tissue and impacted the rest of epidermal function, and even had impacts in basement membrane zone, which then would, of course, impact the dermis. We also found that it was that the abnormality, that any kind of disruption of that barrier would absolutely trigger chronic inflammation, the lymphocytic destructive inflammation. We found that the barrier was critical in the pathophysiology of 13 major skin conditions and diseases, but we were focused on really trying to understand the underlying mechanisms and then focus on the mechanisms to prevent skin diseases that increase as we age, to prevent skin cancer, because I was seeing teenagers with skin cancers, a lot of them in my practice, which is high desert area, farming, ranching community. And so that was our major focus. Well, so as we made these discoveries and we started treating people with actinic keratoses and rosacea and seborrheic dermatitis, other types of dermatitis, a year after we did the clinical studies, which we were then planning on going on into the prescription realm, people were starting to call me and say, "Hey, I need more wrinkle cream." I said, "We didn't give you a wrinkle cream." And they said, "Yeah, but my family said the wrinkles went away." So remembering what Albert Kligman had told me at one time in a meeting with Dr. Stoughton, that he said, always listen to the patient. So we went ahead then and did a study. And sure enough, we found that our products that contained the barrier repair impact, or the and the anti-inflammatory modulation was very effective. As a matter of fact, we were 200 to 300% better than any alpha hydroxy acids, salicylic acid, kynotin. And so that gave me the confidence that we should go into the prescription arena. Our basic research, we found that there were 5 pathways involved in modulating barrier damage, and we needed to upregulate all of those 5 pathways to get optimum barrier repair. And we found that a 3 to 1 to 1 ratio of cholesterol, ceramide, and free fatty acid, of which 50% was linoleic acid, was critical for triggering the barrier repair. Why do we know that? Well, what we found was that when the skin was damaged for any reason, those were the first 3 molecules that were actually synthesized. And within 45 minutes of injury, they were fully being synthesized. We also found that there were 7 pathways of inflammation, and since then, an 8th one was added more recently with the aryl hydroxy receptor pathway. But we found that these 8 pathways of inflammation occurred, and these were pathways that were actually only 2 were affected by antioxidants. The rest were by other anti-inflammatories. And they set up a whole series of cascades that resulted in an imbalance in matrix metalloproteinases and their TIMPs. And so then that was what would trigger the destructive chronic inflammation, usually 2 to 3 weeks after the injury. So then it was, all right, if this really works, then we should test it truly in an aging model. And about that time, right around that time, emollient tretinoin 0.05% was approved. So we went ahead and did a study with a double-blinded prospective controlled clinical trial against tretinoin emollient plus its lactic acid moisturizer. And we used our renewal face cream, and then we also I shouldn't say we, but I came upon the idea that why not use the delivery routes that are already there? In other words, use the pores. Well, we wanted to make sure that the pores were cleaned out well enough, because we were understanding then that there was actually a defect down in the follicle that resulted in what a lot of people see as acne, as keratosis pilaris, etc.
Hyperkeratinization.
And we wanted to penetrate that, so I developed the lytic products as actually a delivery tool to make sure we cleaned out the pores, so we could get optimization of the renewals down deep and have that impact. Well, the fact of the matter is, in a 6-month clinical trial, we found that there was statistically significant increase in glycosaminoglycans in the epidermis of our renewal and lytic compared to the tretinoin and lactic acid. And then we also found that we had a highly statistically significant increase in superiority of dermal rejuvenation and dermal thickening compared to the tretinoin lactic acid combo. And we had no irritation. We had nobody develop any irritation. 40% of the retinoid people developed irritation, indicating
that was the original Renova, right?
Yeah,
the 0.05.
That was the original Renova.
Original Renova.
We basically exploded the concept that you had to have inflammation to get a beneficial regenerative.
Yes,
result.
which has subsequently also been proven by others, but you guys were again ahead of the curve. So tell us what is in Renewal Facial Cream and what is in the lytic line. You know, those ingredients.
Yeah, so
sort of the hero
one of the
ingredients.
things that was really important for us to do was to prevent the development of tachyphylaxis, because the environmental insults and your metabolism continues throughout. And so we found, working with Dr. Stoughton, that if we had 2 molecules blocking each receptor site, we could reduce it by over 90%. So we have a series of the 3 key barrier lipids: cholesterol, ceramide, and linoleic acid, and other bulk fatty acids, in a proper ratio. We found the ratio was really critical, and we were the first ones that actually discover that. So that's one component. Secondly, we have the anti-inflammatory component, which was extracts of multiple herbs, because what we found is the synthetic anti-inflammatories did not block the pathways well enough, and we wanted to actually block all those different pathways. But because that required complex formulations, we went to the herbal arena, where they not only have the actives, which are secondary metabolites, but they also have entourage molecules that help provide stability and delivery. So we have that herbal fraction there. In the renewal, the key ones that we found was the extracts of meadowfoam, flax, also a specific extract of avocado, rosa canina, but one of the big actors is date, and then specific apple extracts and safflower. Now, these extracts have different purposes, and we have different extracts for different specific purposes. So like safflower has a component for free fatty acid, but it also has a component that provides transferosomes for delivery of more difficult molecules. So we use both of those extracts to do that. So when we get our raw materials, we go through an enrichment process, and we only use pharmaceutical grade. And so they're highly purified, but we actually go through an enrichment process, and then we combine it with a third component, which is the delivery component. And that's, we pioneered several drug delivery technologies, and we utilize those. So we actually, in the renewal, we have 3 different delivery systems. So we can get one targeted to the stratum corneum, one to the basal membrane zone, and another to the granular layer and those other middle strata of the epidermis. So it's really not a specific hero ingredient. It really is the combination in the proper ratio, working on all 8 inflammatory pathways, inhibiting those, and activating all 5 of the barrier repair pathways. We also provide the precursor compounds and elements to continue to activate the rejuvenation process. So it's not just putting it on, but we actually stimulate the processes to trigger continual repair.
That's so fascinating. And what about lytic?
Hundreds of experiments.
I know y'all, but you've been working at it for a long time. And lytic, what is it that cleans out the pores, basically.
In the lytic, we actually use a salicylate that has been modified in the sense that we combine it with mineral and fatty acid, so that it produces a not only more complete penetration, but has a penetrating effect that's somewhat slower but very predictable. And by doing that combination, we don't get the irritation with it. And we don't get, we can really target the comedolatic area with it. Yes, it will take off excessive scaling. So we found it was very effective in treating hypertrophic actinic keratoses and many of the inflammatory diseases like Kebner responses from psoriasis and that sort of thing. But it really is targeted primarily for the follicle.
Interesting. Now, you have your own manufacturing facility, right?
Yes.
Which is highly unusual for those who don't know. Even some of the big major cosmetic companies use contract manufacturers. So what motivated you to do that? That's a huge investment and something that
Well,
even
Patty,
big
because
companies
I
don't
had
do.
22 U.S. patents, we were spending millions of dollars policing those patents worldwide. And I came to the point where I thought, you know what, a more efficient way to do it would be to actually have our own manufacturing, because then we can use our patent. Because at that time, when we set this up, we had a number of patent pending technologies, not only for delivery, but also for these the molecules that actually worked on the various receptors for the inflammation and the barrier repair. And so I wanted to make sure we could protect those. And I knew we didn't have the finances, because I'd used all my retirement and every dime I had on this, that I knew we couldn't afford to do the patent protection. And then the other thing was too, that what we found with the herbal ingredients is that there's a wide variation in the quality of the ingredients, in the sense of the amount of the actives and the way it was processed to maintain functionality. And that's one of the big problems with natural products. And so this way we are able to make sure we have the high quality. We assay them all as we get in these raw materials. As I said, we go through an enrichment process. And so we make sure that we know exactly how much actives we have in each particular one of those fractions. And frankly, we had suppliers try to bamboozle us with faulty products, and when we're doing our formulation, I'd get a call, "Doc, you better come and take a look at this formula, say something's wrong," and we could find out that they were doing all kinds of substitutions. And the other thing is, I also have a basic research foundation, and there we're doing a lot of very early herbal work, and we found that in looking at the major suppliers, the differences in potency of the bioactives that we used ranged as high as 1,400% difference in the different suppliers. I mean, there's a huge variation. And then also what we found is that as we were working on delivery systems for these and so on, we found that there was a great difference in the amount of delivery based on the amount of these entourage molecules, which really hasn't gotten the press until just recently. But we've been doing work in entourage molecules for 20 years.
I was going to say,
and
explain
so that's
that.
why we did
Explain that a little bit. I understand it because I've heard of it before when it comes to things like CBD. There's big entourage effect with lots of botanicals. But explain that to the listeners, because that's a concept we don't talk about much.
Yeah, sure. The major active components of herbs that actually give them the name herb, because it's a medicinal botanical, are secondary metabolites. And there's about 50,000 secondary metabolites that have been identified in different plants. But one of the things that we found that we couldn't have an explanation for why tonics would work better. And as we analyzed why the tonics would seem to work better than the purified secondary metabolites, we found that there were other elements and compounds that actually were stabilizing and would improve the interaction so that the different actives within the milieu of the herbal extract didn't damage each other, didn't negate the effect, and so on. Because with these active ingredients, they will, chemistry happens, Patty, and they will undergo chemical reactions. So these molecules that provide that, and I first was exposed to that with ricin, the second most potent weapon of mass destruction. I was extracting that in the 80s, and we were using it for cancer treatment. But I was extracting from castor bean, and I kept thinking, why isn't the ricin, it's so potent, destroying the castor bean? Well, it turns out it was bound to the azelaids. And so that's where I started looking at the azelaids and ended up with the first patent for azelaic acid for treatment of rosacea. And then we found that it also had certain delivery. Some of these azelaids had delivery components as well. And so that's really one of the key ingredient difference between our renewal facial cream and the intensive nourishing cream is we added azelaids that would have impacts on some of these other pathways, and then a couple of peptides that we could deliver as well. So there's a lot of different functionalities in these herbal extracts. And so the standard way of steam distillation actually destroys most of the antioxidants in it, a good portion of it. And so what we really worked on, particularly with the new extraction methodologies we invented in 2019, was to preserve these entourage molecules. And what we found was, as we were doing all these assays against disease state markers, we found actually additional therapeutic functionalities. Well, they actually have impacts on what we believe are going to be other diseases that we're working on for later. So it's very exciting work to see how these things work, but it's a very complex process. And we were so surprised to see the amount of difference in functionality between different commercial carriers, and realized that a lot of it had to do with the manufacturing and the extraction methodology.
So do you make the extracts yourself in your facility, or are you more testing the extracts?
We get pharmaceutical grade extracts from companies that have specific ones, and some of them are made from real small companies. And then when we've identified that there's a particular molecule that we need and the natural extract does not have enough concentration in that, then we will augment with that particular molecule into that. And it's interesting how they're far more stable by doing it this way, so that by having the entourage molecules in the herbal is much better than mixing a number of the synthetics together or putting in these other natural ingredients together. So we found that that's a good base. So we find a lot of using the herbal extracts for the base for other ingredients that are hard to deliver.
Right. So one of the big buzzwords we're hearing a lot about now is like microbiome friendly. And, we know that things that perturb the microbiome upregulate inflammation and cause skin sensitivity and dry skin and all those things. And it would seem like your line would be right in line with that sort of philosophy of nurturing the microbiome and keeping the stratum corneum and the skin barrier happy and healthy. Yeah,
it absolutely does, Patty. It's really the real focus of Epionce is to optimize the epithelium. That's where the name Epionce came from. One chance for an optimum epithelium. And so absolutely, we provide the base for the optimization of microbiome function as well. And, 15 years ago, 10 years ago, we didn't really hear much about the microbiome. But one of the things that was important, all of our anti-inflammatories that we screened had to have impact against pathogenic microbes, whether they be yeast, parasite, and different kinds of bacteria. So that was one of our criteria for the anti-inflammatories. They had to modulate or basically inhibit the disease-causing ones. Well, subsequently, what we found is that actually we also upregulate some of the microbiome, the good microbiome as well. We provide some prebiotic for them, and by providing the proper pH and so on, by optimizing barrier function, we're finding that the good probiotics that are really needed for optimal skin health, we actually upregulate as well.
That's totally fascinating. I think this is an area that we're going to see a lot more and hear a lot more from skincare companies about is the microbiome and how some products that we use affect it in a negative way and others obviously in a positive way. You mentioned prebiotics. Do you use any postbiotics, any of the bacterial lysates in your products, or are they primarily
No, we haven't to
botanicals?
date. I haven't felt the need to do that at this point in time.
Well, if you have
But
the
I follow the
prebiotics,
science very carefully and see if there's something that's actually going to augment. Now, I mentioned earlier that there's an eighth pathway of inflammation that was worked out and published in 2018, and it turns out that when we actually started studying that. That what we have already in the intensive nourishing cream and the renewal products actually modulate that pathway as well. And so we did another series of studies on that and found we have a very good inhibition. Not the most potent inhibition of that eighth pathway, but nevertheless a very good inhibition. And new products coming. There's things down the
the
road.
hits keep coming from you guys.
Exactly right. I
What
mean,
is your most recent
Patty, we have
launch?
done. I'm sorry, Patty. Sorry for interrupting you, but we have done 60 in vivo trials, and we've done more than 20 products, blinded clinical trials, and we have one underway going right now. So it's we'll continue to do that. And one of the reasons I wanted to have my own contract manufacturing or my own manufacturing facility is because it allows me to invent and start early stage of the formulation development so on these products. And I'm able to do that by having my own facility.
Yeah, how many products are in your line? How many
30.
SKUs?
See, it's a little bit less
30
than
something.
30. So
Oh, wow.
right around 30.
And do you distribute only through physicians' offices or are you direct to consumer as well? I think you're direct to consumer now.
We for years, we only distributed through physicians' offices and med spas where they had lasers and a physician overseeing it. But then a couple of years ago, we went to controlled website, and we have a
Mm-hmm.
couple people that sell like Lovely Skin,
Mm-hmm.
and then we also were having so much trouble with diverters, and then that's been one of the biggest problems.
Mm-hmm.
And so we actually went on a preferred program with Amazon last year, and that has really had a big impact in reducing diverters.
Yeah, diversion's a huge problem for
It's a
everybody.
huge problem, and it was like whack-a-mole for a number of years. We spent millions of dollars trying to
Yeah,
stop it on our
you
own.
can't. It's like
Can't do it.
it can't. Yeah, it's
Yeah, yeah,
impossible. Even people have entire organizations to try to stop it, and they can't do it. Well, that's such a fascinating story. We're almost out of time. I know you're kind of a renaissance guy as well in terms of you do a lot of outdoor activities and you to me you're kind of a well-rounded you're sort of a brainy guy and a little bit of a we call ourselves the skincare nerds, who really likes to get down in the weeds and look at it, but
yeah.
you also do a lot of outdoor stuff. How'd you end up in Boise, Idaho? Are you from that area?
No, I actually grew up in the Columbia Gorge, but I've been very active for a long time. Mountain climbing, skiing. I started skiing at 4. I've done 54 peaks over 11,000 feet,
Oh,
including
wow.
23 14ers. And done some triathlons, a lot of cycling and road races and things like that over the years. Been very active, athletic all my life, but one of the reasons I did that, Patty, was because I itched so bad. And
Ah.
so one of the reasons I didn't sleep much is because I had so much itching. So my parents would buy would get phone books from all over the country, and so I would memorize phone books for hours every night to help fall asleep. And then during the day, I found that I itched less if I did a good workout in the cool of the morning or the cool of the evening. So solitary workout, and that would really help. a lot of niching. So a lot of it was necessity. But then I was fascinated with just the challenge. Preparing, my mountain climbing adventures, we trained for 4 months before we'd go. And so it was always great to have an opportunity to do that. And I also did some open water swimming across big lakes and so on. I enjoyed that as well. So,
so interesting.
But,
so interesting. You're living a full life, that's for sure. And you're still at it.
Right. And the other reason we set up in Boise, Idaho was frankly that I wanted to be close to my practice. My practice is 60 miles west from here. But I also wanted, we have really good quality people for work ethic. And I wanted to make sure that the products that I developed, my patients could afford. Because at the end of the day, the whole reason I did Epionce was to make me a better doctor. Keep in mind, when I went into practice, the gold standards for treatment of dermatitis had a 56% success rate with Lidex. And I felt we could do better than that. And that was one of the stimulators. And so I wanted to make sure that our cost of goods was such that we could be able to have cost-effective products for people in my practice, and so that was really
that was your motivation.
Yeah, that was my motivation. So we defied a lot of paradigms to develop Epionce and bring the product to the market. But I mean, Patty, how many people are able to start with a theory, you do basic research for nearly a decade, and then make products for several years, develop product development for several years, and then put it in clinical trials. It works. Then you introduce it in the market and people love it. I mean, how many people get that opportunity in life?
That's a home run for sure. Home run in our business. And you helped all your patients as well.
Oh, absolutely. Absolutely. These are game changers for patients.
Yeah.
As far as reducing side effects of prescription products.
What about post-procedure, Carl? Have you looked at these products post-procedure?
Yes, we have. We actually did a study with a cream, or it's a combination that we use after procedures. And we showed, we treated for 4 days. We compared it with the Aquaphor and the acetic acid soaks, and we treated for 4 days. At the end of day one, the Aquaphor acetic acid soak had performed better, but at day two, we were comparable. At day 4, we were statistically superior in reducing all the inflammation and so on. But what was interesting, we then did no treatment for the next 4 weeks. We analyzed the patients then at 4 weeks on 7 anti-aging or aging parameters, photoaging parameters, and we found statistical significant superiority or highly statistically significant superiority of our kit compared to the standard treatment. So in other words, we were activating repair processes that
kept
not
going.
Just exactly right, and kept it going for 4 weeks. And so we've done, that's the kind of study. And we had a feeling that was going to happen because when we did a study against a prescription lachydrin, and again, we treated for 4 days, we continued to have statistically superior hydration and functionality, even 10 days after stopping treatment.
Totally fascinating. Well, I'm going to wrap it because we're a little bit over time, but I'm going to tell the audience that we're super excited that Dr. Carl is going to be at the Science of Skincare Summit this year. Our meeting is going to be held in Austin, Texas, November 8th through the 10th at the Fairmont Hotel. So for all the rest of you skincare nerds out there, join Carl and I. We're going to have a good time for sure, and we look forward to hearing your talk, which I think is going to be on botanicals and botanical extracts. And thanks for joining me today, and especially for joining us at the meeting this year. It's always great to see you.
Thank you so much, Patty. It's such a pleasure. A real pleasure. And if you want to have me on again, I'll gladly welcome the opportunity.
I think we may have more to talk about. Yeah. Thanks, everybody. Have a great day.
Thank you.
Bye-bye.